Hi, everyone. Welcome to our last session of the year. Feels crazy that we are here, but we are. Good news is, at this point, you've put in all of the work possible. So I have some last-minute tips and tricks for you, but just go confidently knowing that tomorrow you have your exam. You're going to be absolutely wonderful humans. Yeah, you can come out on the other side a better version of yourself and having successfully made it through. So I see we already have a good amount of questions. That's awesome. I'm excited to answer all of them. I just wanted to go through a few last minute tips and tricks before we jump in, and then I'll pull up whatever necessary slides to answer your questions. So just my go-to strategies that I highly recommend for multiple choice. I'm always going to tell you, and if you've been listening for the past however long, you know this already, but attempt grid-ins first. Some will be easy, some will be hard. Skip the hard ones, answer the easy ones, and move on. Just because you can answer something doesn't mean you should. So that's the strategy there. The reason I tell you to do them first is because you can't guess on them, because you can't guess a random number. Multiple choice, the actual questions, not the grid-ins, they range from easy to medium to hard to medium to easy, so don't get caught up in the weeds in the middle, and make sure that you're giving yourself enough time to get to some of those easy multiple choice at the end. Go with your gut, trust yourself. At this point, if you're not going in with confidence, what are you doing? Because there's nothing you can do about what you don't know. So if you see a problem and you don't know it, chances are half of the 240,000 students who are taking this exam tomorrow also don't know it. You can't stress about how you don't know it because that doesn't fix anything. Yeah, so don't stress too much about that, and don't spend more than a minute or two per question. It works out to like 1 minute and 20 seconds per question is what you're allotted. So if you're spending a lot more time than that on that, just know that you're taking away time then from other questions that need it. For free response, answer the short questions first and then the long questions. So save 1 and 2 for last. Long responses can be answered out of order. I mean, anything can be answered out of order. I just highlighted it for the long responses, meaning like you could do part D and then part B and never even get to A or C if you run out of time, but at least you can prioritize the ones that are easier to answer. There are always some parts that are going to be easier than others. Be concise. You shouldn't be writing more than a page for the long FRQs. If you're writing more than that, you probably don't know what you're talking about. So, keep it quick. And then be specific. Yeah, questions 1 and 2 are worth a lot more points, but students usually waste a lot more time on them. So Drew, what I've talked to probably 15 different readers who read your exams and grade them, and they say that very frequently they can tell that students spent probably 45 minutes, which is half your time, on a response that they end up only getting a point or 2 on. So if you can prioritize and get those short ones out of the way, then your long free responses, 1, you don't have to time manage because whatever time you have left you can devote to those. And 2, you're usually more concise when you have less time. If you're trying to time manage, you'll oftentimes waste too much time on those long free responses. Cool. They're definitely not going to overload you on plants. Sorry, I hate to break it to you, but plants are a very small part of this exam. Sorry. Yeah, so questions 1 and 2 are each worth 10 points. Questions 3, 4, and 5 are worth 4 points, and then questions 6, 7, and 8 are worth 3 points. But you can get a lot of bang for your buck on those shorter questions, and they're also really quick, so you can get through them really quickly, and then give yourself more time for those long free responses at the end. But students tend to do pretty poorly on the long free responses. We went through a long free response yesterday that the average was a 2.8, I think it was, out of 10. So yeah, they're oftentimes really hard for students to get a lot of points on. So that's that. Okay. Lytic and lysogenic cycle. So I'm jumping into our questions. We have 25 already, so hopefully I can get through most of them. Lytic and lysogenic cycle, certainly I can go through that. Let me pull up a picture quick. You guys have to bear with me. I mean, I know most of you have been bearing with me for all of these Cram sessions, but I do need to find the supporting document so that I can show you the pictures that will help you understand this better. So it just takes a second for me to switch over. What I can say is while I'm finding these, so lytic is going to be where the cell is lysing, and lysogenic is actually going to be where, oh my gosh, where did my folder just go? Where the cell is kind of like in a state of kind of hibernation. So I like to always talk about cold sores with this because it's the herpes virus and everyone's familiar with cold sores. If you have cold sores, you have them forever, but it's usually just in the lysogenic phase, meaning that it's not presenting itself, it's not active at that time. But when it goes into lytic phase, which is when the DNA is actually going to remove itself from the host's DNA and it's going to start producing more virus, that's when you actually see the cold sore, because that's your body actually having that reaction, and that virus is actually killing those cells around your mouth, which is why you see that. Let's see, bacteria and viruses. It's fun to just multitask and talk at you guys while trying to find things. But let's see, I'll pull up the necessary slide, but hopefully everything that you said so far makes, or what everything I've said so far makes sense. Where are you? I can also put this presentation in here. So there's that. And whenever this decides to load, I think my internet gets overloaded when I try to open all of these things at once. I will show you my diagram and go through it briefly. But there's also other helpful information here that you can look through. Bacteria and viruses are my personal favorite. Unfortunately, they're never that large of a part on the exam, which is unfortunate because they're awesome. Okay, let's see. Here we go. Okay, so now you can see lytic and lysogenic. T cells are not part of the humoral response for Sarah, who's ever asking on the left-hand side. Okay, so lytic cycle you can see on the left. So when lytic cycle is induced, and this could be any number of things. For cold sores, it's oftentimes stress, or if you're sick with another infection, your body's kind of distracted or weakened. And you can see that this DNA here, that blue chunk of DNA comes out. It gets replicated by the host's enzymes because they are not capable of replicating their own DNA. They don't have the proteins necessary to do that. That's what makes viruses technically non-living, according to many scientists. You can see that then the cell lyses, releases these phages. And again, if you think of like a very active cold sore, you have that red swelling. That's because those cells are being destroyed by these viruses. On the other side, which is what makes viruses so dangerous, viruses are also able to integrate their DNA into the host cell, which is what you see on the right-hand side. And what's happening there, then the cell is replicating as normal, but they're replicating with the viral DNA in them as well. So that's never leaving. And it's going to continue to stay in that cell until it switches into the lytic cycle again if your body is under stress or something along those lines. But that's how viruses can lay dormant for long, long periods of time. Like if you've heard of shingles, which is a manifestation of chickenpox, that stays dormant for, you know, could be like 50 years, and then it goes into the lytic cycle for whatever reason. So those are those two. Hopefully that answered that question. Okay. All right, sex-linked traits and Hardy-Weinberg. Sure. I'm actually going to show you an example of a grid-in question that I pulled up last night that talks about sex-linked traits in order to talk about it so that we can also, you know, tag team and practice some grid-ins as well. That should be beneficial to everyone. And then we can talk about Hardy-Weinberg briefly as well. That's a good one. Okay. I can actually, that's a grid-in as well. So this is easy. We'll do a little bit of mathematics practice. Let me get our other slide up. So if you have questions about what I'm talking about currently, like what question I'm currently on, you can ask those in the right-hand side chat box. If you have like a brand new question, you need to ask it in the chain of questions. Otherwise, I probably won't get to it. But if you have like a question about what I'm talking about right now, so sex-linked traits, I would be more than happy to answer that if you answered on the right-hand side chat box. I'll try to monitor that frequently. Who clicked right to it? Okay. Cool. So here is an example of a sex-linked trait. So what you need to know about sex-linked traits, and I like this question because it'll also talk about autosomal traits, so we can review a lot of that. Here we go. Hopefully your screen pops up shortly here. It still says loading on mine. Goodness gracious. There's so many of us on here right now. Parts of the slide didn't load. What is going on? Okay, here we go. So it says in fruit flies, the allele for vestigial wings is recessive to the allele for wild type wings, and the allele for white eyes is recessive to the allele for red eyes. The gene controlling wing type is carried on an autosome, whereas the gene controlling eye color is carried on the X chromosome. Okay, so this is super important. Autosome is an important vocabulary word that you should be familiar with. Basically, all that means is it's chromosomes 1 through 22. So all of the normal traits that you think of when we're like big W, little w, or like capital A, lowercase a, all of those things, those are autosomes. So those are carried on our regular chromosomes. So you're no more likely to have a trait or not have a trait if you are male or female. It doesn't matter. It makes no difference. Okay. So again, 95% of the traits that we always talk about are going to be autosomal. So then the gene controlling eye color in this instance is on the X chromosome. So this is going to be sex linked. And what makes it different is that males only have one X chromosome and females have two. So for females, this manifests itself, the same as any autosomal trait, right? You can have two versions. You can be dominant recessive, or recessive. So homozygous dominant, heterozygous, homozygous recessive. For males, that is not the case, because they only have one version of X. So if they get the dominant version, then they're dominant. If they get the recessive version, then they're recessive. But there's like nothing to cover it up. There's not a heterozygous option here because they only have that X chromosome. Okay, so this question then says a true breeding female. So this is a reminder, true breeding means homozygous. So a true breeding female with wild type wings. And we know wings are going to be, I wish I could write on these, but I can't. I can only write on, oh, I wonder if I can add a note. Oh, no. That didn't go according to plan. Okay, never mind. I can only do that on actual PowerPoint, not Google Slides. So I'm just going to talk you through this, and hopefully that's good enough for everyone. So because we know true breeding wild type wings, we know that the female, I'll trade this in the chat box. It's going to be uppercase W for wings. I used W's because the dominant trait is wild type. So usually they use the letter of the dominant trait there. So she's wild type for wings. We know wings are autosomal. And then she has white eyes. And we see that the gene controlling eye color is on the X chromosome and that white is recessive. So mom has two X's, right? And she's showing the recessive phenotype. So I would write that X little e, usually it's a superscript, but I can't do that obviously in this box, X little e. Then with dad, dad is vestigial wings, so he's homozygous recessive then, so lowercase w. And then he has red eyes, okay? So he only has one X. He has the dominant trait for it. Should have used R's, not E's, but that's okay. So he has X with that dominant E on it, and then Y. Okay, so that's our genotypes to start. And so now we need to figure out what's going to happen for our offspring. So if we cross these two, it says what proportion of the offspring are expected to be males with wild type wings and white eyes. So there's a lot of things going on in that we need to unpack briefly. Now, a lot of my students at this point would try to make a dihybrid cross, and you certainly can, but you certainly don't have to. So I'm going to just like walk you through kind of my cheat sheet. So let's look at the first trait. So if you look at these two traits separately, it's honestly going to make your life a whole lot easier. The first thing you should notice is that every single offspring are going to have wild type wings for this couple, right? Because we're crossing a dominant, a homozygous dominant with a homozygous recessive individual. So all of our offspring are going to be heterozygous. All of our offspring are going to have wild type wings. So we can essentially disregard that trait for now because we know 100% of the offspring are going to have those wings. So now you can do a Punnett square, which I can't draw, but you could draw on your own between the sex-linked traits. So crossing the X superscript small e, X superscript small e with the X big E and Y. Okay, so what you'll find is on the top of your table, you should have two females that are going to be X big E, X little e. Okay. And then on the bottom of that, if you cross those, you'll notice that mom is going to give all of her baby boys her X with that recessive lowercase e, and dad's going to give all his boys a Y. That's why they're boys, okay? So it doesn't matter that dad has that dominant allele. None of his boys are going to see that because they get the Y from dad. So all of our offspring then, all of our males are going to be wild type with white eyes, because you can see that there 100% of males have X little EY. 100% of females are heterozygotes, but we don't care about them in this question. And then 100% of males also have wild type wings. So your answer here, it says give your answer as a fraction or a decimal value between 0 and 1 would be 1, because 100% of the males would look that way. So this is a good example of a grid-in, as well as talking about sex-linked traits, as well as talking about autosomes and Punnett squares and all that good stuff. Hopefully that's helpful. Hopefully that makes a little bit more sense for you. Let's look at a Hardy-Weinberg practice problem then. Here we go, question 2 down here. Stop moving my mouse so that you guys can actually see it. So I like this question. The capital and lowercase, Anusha, so the capital is going to be the dominant trait and the lowercase is going to be the recessive trait. Why wouldn't it be 50%? Because we're only looking at males. Only males. So females, like half of the offspring are going to be wild type wings and white eyes, but 100% of males are going to be wild type wings and white eyes because males are XY, right? So the females are the ones that are XX. So 100% of our females are going to be carriers here, but they're going to have red eyes. And 100% of the males. So our males are the XY ones. So the only XY iteration that we have is the recessive one. Is everyone good with that now? It says total population of males. I'll go back to it. Right here. What proportion of the offspring are expected to be males with wild type wings and white eyes? It's asked. I know there's a lot of confusion here. What proportion of the offspring are expected to be males? Oh no, I'm so sorry. You guys are right. It is 50%. Yes, I thought it said what, see, and this is why, yeah, sometimes you can, yeah, the males are the X subset E and Y. So 50% of the total offspring are going to be males with wild type wings and white eyes, but 100% of males are going to have wild type wings and white eyes. But you're right. You're absolutely right. So that's a really tough question. It's also a lot of work for a grid-in. So I saw some people said they would skip that. I would skip it as well. But you're 100% right. I completely messed up there and misread that. So the answer would be 0.5. Yep. Because it's talking about the proportion of all offspring. You guys are 100% correct. Sorry about that. Sometimes my brain, this happens to me in class too. I'm sure your teachers do the same things. My students are like, you messed up. I'm like, oh, yeah, it happens. But yeah, you guys are 100% correct. It's okay. Everyone's close to going crazy at this point. All right, so let's take a look at question 2. A population of flamingos have pink and white feathers. Pink is dominant to white. If 74% of the population has pink feathers, how many are heterozygous? So I think I reviewed this question perhaps on Wednesday. I can't remember because I also am forgetting what I have and have not talked about. But the main thing to note here is that 74% of the population is pink and pink is dominant. So this number is actually completely not usable to us. So what my students would love to do at this point is to set 0.04 equal to P, or set 0.74 equal to P squared. And neither of those is accurate, and I can explain why for both. We can't set anything equal to P or Q because we're not talking about allele frequencies. And this says this on your equation sheet, but only the frequency of the allele, the dominant allele or recessive allele, can be set equal to P or Q. And we can't set this equal to P squared because we don't know that all of these pink flamingos are homozygous dominant. Okay, so 0.74 is actually equal to P squared plus 2PQ. Oh yeah, Friday. Okay, perfect. Thanks. So sorry for whoever asked if I'm repeating this, but all of you are 100% correct. Q squared is then equal to 0.26. So you always want to go with recessive because recessive phenotype is equal to the recessive genotype. Dominant phenotype is not equal to the dominant genotype because you have heterozygous and homozygous dominant mixed there. Yeah, so Q squared is going to be equal to 0.26. So you go ahead and square root that, and you should get that Q is equal to 0.51. If Q is equal to 0.51, then you find out that P is equal to 0.49, right? Because P plus Q equals 1. And then you can do 2PQ and plug all of those in, and you should get 0.50 or 0.499, depending on how you round. So that's just a good example of why you cannot ever use the dominant phenotype, because it doesn't give you enough information. You actually don't know what that means. For question 1, you're absolutely right. Number 3 is correct. The amniotic egg is what alligators and manatees are going to have that salamanders do not. Okay, cool. All right, sorry, we spent a good chunk of time on that question, but it's a good one. Okay, next. Ooh, 17 votes. Sorry, I'm going to skip ahead. All right, how would you describe the Krebs cycle and the Calvin cycle in a few words if we were asked on the FRQ section? Oh wait, I'm sorry. I want to make sure that I've answered these questions for Drew and Raj. If it said the frequency, of the dominant allele, if it said the frequency of the pink flamingos, then if it's still talking about flamingos or individuals, but if it says the frequency of the dominant allele, then you can set it equal to P. Yep. Yeah, so there's that. Okay. What are the different ways they can ask for? They can ask you for anything in Hardy-Weinberg. They can ask you for a frequency of an allele. They can ask you for the frequency of homozygous dominant. They can ask you for frequency of heterozygous. They can ask you for any of those things. Yeah, so they can ask you a lot of different ways. But they'll either ask you for the frequency at the end, and you'll plug that in as a decimal, or they'll ask you for a percentage at the end, and you'll plug it in as a percent, or they'll ask you for the total number of individuals, in which case you would need to multiply your frequency or your percent by the total population that you're given. Yeah, any of those ways. Okay. How would you describe the Krebs cycle and the Calvin cycle in a few words if you were asked on the FRQ section? So they're never just going to ask you to explain those things on their own. So if you guys have looked at, I think it was it was last year's. No, it wasn't. It was 2015, I think, or 2016. I'm losing my mind. The free response question that talks about cellular respiration, it kind of gives you a good idea of how you needed to talk about Krebs. But here are what I would say are the most important things that you need to know about these two. So Krebs cycle, the most important thing that happens here, so we're breaking down acetyl-CoA into carbon dioxide, which is just a toxic byproduct. But the real important thing is that when we're breaking bonds, we're releasing electrons, and those electrons are getting picked up by electron carriers. So that's really the most important thing about Krebs cycle. We also make a little bit of ATP, 2-ATP, but way more important than that, we make lots and lots of electron carriers because we're breaking lots of bonds. Anytime we break bonds, we're going to release some energy, but we're also going to release some electrons, and then those electrons are going to be put to work for us in the electron transport gene. For Calvin cycle, so Calvin cycle is a really important process. It's kind of the opposite of the Krebs cycle, but it's not the actual opposite. It's going to happen in photosynthesis, obviously. And we're going from carbon dioxide to glucose, or glyceraldehyde 3-phosphate, if we want to be really technical, G3P, because that's going to be the precursor to glucose, but also starch and cellulose and all those types of things. So what happens here is we have carbon fixation, which is really awesome. Plants are capable of taking CO2 out of the air and turning it into an organic compound. That's done by an enzyme called rubisco. Okay, but then most importantly, we're relying on our light reactions to produce a ton of ATP and electron carriers, right? So those electron carriers come in and they drop off electrons. We also drop off ATP, and we're able to use those electrons to form more bonds. Because if we're going from CO2 to something complex like G3P or glucose, we're going to need lots of electrons for that. And we're also going to need energy to make that happen. So we're also going to do the lovely job of recycling those electron carriers and the ATP into ADP, and we send them back to the light reactions to be refilled. Remember that those things are limiting reagents. If you're taking chemistry, you know about that. So we need to go ahead and recycle them, and so the Calvin cycle does that quite nicely. So if you can remember those overall principles, like what the Calvin cycle does and why it depends on the light reactions, and what the Krebs cycle does and why we depend on it, you'll be in good shape. But they shouldn't just ask you to like straight up explain it. They'll ask you something specific, like what does this electron do or something like that, and you can kind of walk through the process like that. Okay. Operons, would love to go through some operons. Let me pull up a PowerPoint slide. So give me just a second. Operons are cool, and their names make a whole lot of sense. So I'm just going to go over prokaryotic regulation, the other ones that do operons. Ours is a little bit more complicated, and you don't need to be as familiar with it. But I'll pull up my gene regulation PowerPoint for you, and I'll also post that in the chat. but there's a lot, and I just kind of said this, but there's a lot in a name. So the fact that we have like inducible and repressible operons is actually very important, because the name tells you exactly what they do and if they're normally on or normally off. Okay. Pull up an example of both and we can zoom through them very quickly. My gosh, we're almost halfway through our time already. Boy, oh boy. Come on, Mr. Computer. Okay, the Operon. Here we go. All right. Let me post this in here because there's lots of good information in this PowerPoint that you guys might like. And then I'll make this full screen and we shall chat. Okay, so we're going to start by talking about an inducible operon. So inducible means able to be induced, right? Able to be turned on. So what that means is it's normally off. Okay, so a couple key parts to an operon that you can see here. We have a regulatory gene, we have a promoter region, we have an operator. So the regulatory gene is upstream, it's away from the genes that we're actually trying to transcribe, and it makes a regulator protein. In this case, with an inducible operon, it makes a nice active repressor that goes ahead and binds to the operator, which blocks RNA polymerase. So if you think of RNA polymerase as like a train on the tracks, we've got a big old tree in the tracks because that repressor is bound to the operator. So the train can't pass, we can't transcribe or translate any genes past this blockage, and so therefore we're not going to be making whatever RNA or protein is supposed to be made here. So this is off. I always think of inducible operons like the lights in your house. You know, you want them to be on when you're there, but you want them to be off when you're not in order to save energy. So inducible operons are for things that we don't need all the time. The repressor is made by the regulatory gene. So the regulatory gene transcribes and translates the repressor, which you can see on the left-hand side of the screen. Okay, so we don't need these things all the time, and so we're going to turn them off when we don't need them so that we don't waste energy. And by we, I mean prokaryotes, because we do not do this. We do regulate transcription, but not in this exact way, because we don't have our genes linked quite like this. So this is the inducible operon when it's turned on. So this particular example is the lac operon, and it is the most common example of an inducible operon, so you'll probably see it. If they're going to put an operon on there, they're going to put either the lac or trip operon on there. So what you can see here is that when lactose is present, so real quick backstory, lactose is a complex sugar. Bacteria would like to break it down, but only if there's like not glucose available, because glucose is way easier to break down. This is way more complex. So if lactose is present, so the E. coli that live in your intestines, you drink a milkshake, all of a sudden they have all this lactose to break down. What's going to happen is the lactose basically clogs up the repressor. You can see that there with that green allolactose, which is just an isomer of lactose binding to that protein, clogging it up and allowing it to actually be removed from the operator region. What you can see then is RNA polymerase is able to now finally go past like that train on the tracks and transcribe and translate all these different enzymes that are responsible for breaking down lactose. So we don't want to break down lactose. Again, we don't want to make these enzymes if there's no lactose present. So if you never eat lactose, these enzymes will never be made by the bacteria in your stomach because it's a waste of energy, but it's only going to be made when lactose is present. Now, the cool thing is these enzymes then will obviously go ahead and break down lactose. When they've broken down all the lactose, the repressor will become active again because there will be nothing to bind to it anymore because we've broken everything down. Then the repressor fits back on and we get turned back off. Okay, so inducible is going to be for things that we don't need all the time, that we only want to make when something is present or when there's some sort of need. But again, it's like lights in your house. So you can turn them off when you're not there. This is all just with bacteria, yes. Okay. So then repressible is going to be the opposite of this. Repressible, able to be repressed, which means it's going to be naturally on. So this is going to be for things that we need all the time. So the classic example here is tryptophan, the trip operon. So we need tryptophan all the time. Tryptophan is an amino acid. If we don't make it, if we don't have it, then we're not able to make a lot of our proteins because we need those amino acids to make our proteins. So here the difference is our regulatory gene naturally makes an inactive repressor, which you can see on the left-hand side. So RNA polymerase binds and goes ahead and transcribes and translates these different units that make up enzymes that make up tryptophan constantly, right? Unless something else is there. So what happens then when we already have tryptophan? So let's say you eat turkey. Turkey is loaded with tryptophan. And now the E. coli in your stomach don't need to make tryptophan anymore for a little bit because they have a naturally occurring supply of it. Well, tryptophan actually acts as a co-repressor and activates this repressor and turns it off. So in a repressible operon, the thing that we're making acts as its own co-repressor, and then in an inducible operon, the thing that we're trying to break down acts as an inducer. It's actually gonna turn this thing on. Okay, so this thing is only gonna turn off if we already have tryptophan present because we don't want to waste energy making more tryptophan. But at the same time, it's normally going to be on because we normally need tryptophan. It's one of those things we can't, not just like oh, we can live without this kind of thing. A co-repressor is just something that is going to activate the repressor. So these aren't like perfect examples of feedback loops, so I wouldn't necessarily view them as such, except for that like when lactose is present, then we break it down. So if anything, it's going to be negative. It's not going to be positive, but it's also not really a great example of a feedback loop because we don't really have, yeah, we're not really trying to maintain an equilibrium here except for breaking down lactose. Yep. So there's your operons for you. So hopefully that makes a little bit more sense about them. Again, if you think about what the words actually mean, repressible and inducible, then you have less memorizing to do, and you can more just think critically about what you have here and why. I wouldn't say that the lac operon is positive control because we're not increasing a stimulus. Yeah. Okay, similarities between cellular respiration and photosynthesis. Okay, so cellular respiration, I mean, the equations are inverses of one another, but that doesn't mean that the equation or that the formula is just running in reverse. But if you remember one of them, then you remember the equation for the other because it's just the reverse. There aren't a whole ton of similarities except for that both have electron carriers and both make ATP. Both do have chemiosmosis, both do have ATP synthase, so I guess there actually are more similarities than I'm letting on to. Both have an electron transport chain that's going to create a concentration gradient through chemiosmosis. Then we're going to make ATP. Yeah, electron carriers are going to be used in two different ways. In photosynthesis, electron carriers are going to be used to donate electrons to make bonds, so as we make glucose. And in cellular respiration, we're going to use those electrons in order to create a concentration gradient to make ATP. So those are some similarities between those two processes, but one is making glucose and one is breaking down glucose. So there's a lot of differences in there as well. You do not need to know numbers. An electron carrier would be like NADH, FADH2, NADPH. All of those are electron carriers. Chemiosmosis is just the movement of ions from high concentration to low concentration. So we create a concentration gradient of hydrogen ions in both cellular respiration and photosynthesis. So we pump hydrogen ions against their concentration gradient using the power of an electron. And then when those hydrogen ions come back through ATP synthase from a high concentration to a low concentration, we're able to harness that energy kind of like a windmill or a water wheel. We can actually use that energy of that flow of ions in order to phosphorylate ADP into ATP. Electron carriers carry electrons and then they drop them off other places. So they're just like little shuttles. That's really all they do. The electron transport chains in photosynthesis and cellular respiration are actually exactly the same. They're both making ATP. Yeah, I mean, there's not oxygen at the end of the electron transport chain in photosynthesis. The electrons end up on the photosystems around an electron carrier, and there is an oxygen at the end of the electron transport chain for cellular respiration. But the actual process where we're pumping hydrogens across the membrane and then letting them come back through is going to be the same. Substrate level phosphorylation is just adding a phosphate to something. So like ADP, adenosine diphosphate, gets a phosphate added to it through substrate level phosphorylation in order to become ATP. Okay. Whew. Lots of good questions, y'all. You're asking really good questions. I'm trying to make sure I got through all of them. Cool. All right. Quick review of the immune system. Oh, sorry. The oxygen that's given off by plants and received by animals is used where? So it's given off by plants in the electron transport chain when oxygen is donating an electron, and it's used by animals at the end of the electron transport chain. Yeah. Oxidative phosphorylation is going to be at the whole process of the electron transport chain. So the use of oxygen at the end of the electron transport chain, and then oh, sorry, I got to close the door really quick. And then the phosphorylation of ADP through ATP synthase. Let it out there. Okay. Cyclic and non-cyclic, that's in photosynthesis, and I would totally not stress about that. Okay, cool. Oxidative ATP, yeah, all these things are true. Forward reactions are not always exothermic. It depends on the reaction. Okay, cool. I'm going to jump back into some of these questions unless we have a few others, but hopefully those were helpful answers. Okay, immune system, innate and acquired. Yes, let me pull up a PowerPoint real quick. Immune system is actually pretty straightforward, so don't be too stressed about it. Yesterday we looked at the 2016, I think it was, free response, and they have a really good immune system question 8, that you can take a peek at. We pulled it up yesterday. Yeah. Immune system, and I'll post the link to these slides as well so that you guys can view them, even if I don't get through everything right now on your own time. Not that there's a ton of time, but you can certainly review on your time. Okay. Ooh, is that coming up? It says you are screen sharing, but it doesn't show anything. That's cool. All right, let's try this again. I really hate technology. Substrate, you're right. Substrate level phosphorylation is just ADP plus P. You guys got it. No worries. Okay, here we go. Hopefully this works now. Come on. Okay, lovely. All right, so we'll focus here because this is a decent example of like non-specific versus specific, is what the question was asking. Come on. Oh my goodness. I must know that everyone's stressed about the exam tomorrow because it's just working extra super duper slow. Okay, non-specific. So innate immunity is our first and second line of defense. You're born with it, doesn't have memory. It fights the same speed every single time, and it's going to be really quick. So those are things that you need to know about innate immunity. So the first line of defense is all of your outer coverings. So mucus, skin, secretions, coughing, sneezing, hair, anything like that. Those are all first line of defenses. And if you think about it, like think about what your skin does for you every day. It's protecting you from bajillions of pathogens just by being skin. If your body was exposed just to the outside world, you would get sick constantly. That's first line of defense. Again, you're born with it. It's going to treat all pathogens the same. It doesn't have like specific recognition, and it doesn't have memory. Second line of defense is going to be if we get past the first one. So these make sense, like if you think about first, second, and third line of defense in like a battle or whatever. Now we're inside the body, and we have a couple different things. So we have phagocytic leukocytes. So those are going to be your white blood cells that are going to eat non-discriminately anything that's not you. We have antimicrobial proteins, which can puncture holes in microbes, which is cool. The inflammatory response, so swelling and heat, and then fever, all of those things. Like a fever is actually your body's second line of defense. It's trying to heat your body up to actually kill the pathogens that are in it. So that's actually a defense from your body, but it's spurred by the pathogens that are inside of you. So again, all those things are going to treat pathogens the same. They're not going to be any faster the second time they meet something. It's going to be the same the whole time. And then our third line of defense is the one that's the specific one that has memory, where we have make antibodies, we have lymphocytes, we have those T and B cells coming into play there. MHC is going to be the proteins on the surface of the cells that recognize you versus not you. Let's see. Okay, so let's talk about the third line then. So here's a good example of this. So we have a pathogen and we have a B cell that recognizes it. So you need to know that the antigen or the projection on the surface of the cell is going to match up specifically with another cell type and or with the B cell's little arms there. And so an APC is an antigen presenting cell, and MHC is the proteins on the surface of your cell that mark you as you and not you as not you. So if they recognize an MHC that's not yours, it's you don't need to know all the specifics. It's a major histone complex. It's just a protein surface marker. Your B and T cells actually learn when they're maturing. They learn who you are and who you are not. So they learn your MHC marker. It's like a tag that's just unique to you, which is pretty cool. And so your cells will recognize you as you. This is why like organ transplants can go wrong because the organ that you're transplanted has different MHC markers that your body can actually fight against. Okay, so what's happening here is we have a pathogen entering, we have a B cell recognizing it, and again, it's going to be very specific how that recognition occurs. The specific projection is going to match up with a specific antigen on the pathogen, and a couple things will happen there. So helper T cells will also help bind to these things in order to send out the alarm that we are under attack. Activated B cells will have some plasma cells produce antibodies, and antibodies can neutralize our pathogens. Macrophages are nonspecific, you're correct. We also form some memory cells, and those memory cells are then going to be stored so that we can have a much quicker reaction the second time around with the same pathogen. And then we have T cells that are going to actually kill any infected cells. Yeah. So let me show you that again. So here's MHC. A self marker MHC is how you identify self versus not you. So again, all of your T and B cells learn this when they're maturing. And then we can see, so here are our T cells. So there we have the helper T cells that are going to send the alarm, but also killer T cells or cytotoxic T cells are going to be able to bind to infected cells and actually kill them. They puncture them with a protein. They basically puncture a hole in the membrane, which is going to have those cells go through apoptosis. They're going to pop, and then we no longer have the infected cell. Okay, lots of other information here, but I know people are stressed and want me to get through as much as possible. You can take a look at this last diagram. This kind of shows you what B cells do versus T cells. So B cells are the humoral response. They're in the humors of the body. I always remember be funny, like humoral, like humorous, be funny. You should always be funny. And then T cells are going to be the cellular response. So they're going to take care of the infected cells, whereas the B cells are going to bind free pathogen. They can also be antigen presenting proteins, antigen presenting cells, so they can eat up a pathogen and then present it on their surface, which is how the helper T cell recognizes them. T cells don't produce antibodies. B cells produce antibodies, specifically the plasma cells. T cells are going to either send out an alarm. If they're a helper T cell, they'll send out a scheme of chemicals, or they're going to actually go and attack cells. Yes, both T and B cells have memory, but B cells are the ones that produce antibodies. Okay. Yep, so there's memory B cells, there's memory T cells. You can see those on this slide. B cells are the ones that make antibodies. You can see that on the left-hand side. And then T cells are going to fight infected cells. So antibodies are going to attack like free-floating pathogens. So if we go back a few slides, I can show you that. Here we go. So this is what antibodies do. So they're not going to kill infected cells for the most part, although in some circumstances they will, as you can see on the right. But for the most part, they're just going to kind of attack bacteria and mark them to be eaten by a macrophage. And cytotoxic T cells are going to attack infected cells. So like your own cells that get infected, they can no longer survive, right? We need to kill them. So they're going to be the ones that are killing any infected cells because we need somebody to take care of both of them. So B cells are going to attack the free pathogen in the blood, and T cells are going to attack infected cells. All right. Hopefully that helped a little bit. I know that we're going through a lot right now and we're jumping all around, but you guys are doing great. All right. Okay, looks like we have a couple up next. Oh, the lymphatic. So those are your lymph nodes. So those are going to be, that's what we're talking about right now. So actually I can go back to a picture really quick, and then I promise I'll go through that cladogram question. So your lymphatic vessels are going to be the lymph. They produce lymph. And this is how your B and T cells travel through your body. So that's the lymphatic. So everything that we just talked about is the lymphatic system. It's just the fluids that your T and B cells travel through. And then the lymph nodes, right, which you don't need to know all the specifics of, but the lymph nodes, which are all over, like in your armpits and your neck, that's what your doctor feels for to see if they're swollen. That's actually where T cells go when they find like a new something that you're under attack with. They go and they like go through like a library of other T and B cells to see if we've seen this before and if we can fight it. I make it sound like it's like a really intense battle, and it is, but that's what your lymphatic system is. So everything that we just talked about with B and T cells, that's the extent of what you would need to know about your lymphatic system. Okay, let's talk about the FRQ from 2018. So I'm going to pull that up really quick, and I will share it with you guys as well. Okay. I will pull it up. I've switched things so many times. And it looks like some of these other questions are shorter, so hopefully I can zoom through a few before I have to say goodbye to you guys. While I'm pulling this up, just remember that you are great and you've done all the hard work. And if you're panicking, don't, because no good comes from that. There's nothing you can do at this point, unfortunately / fortunately, depending on how you want to think about it. But you've done your best, and you've done the best you can do. And now you just kind of have to go forth with confidence, because again, there's no point in stressing about something that you can't do anything about anymore. And at this point, the deed is done. So you guys are going to be great. All right. So question 1, polar bears are highly adapted for life in cold climates. All right, so it looks like we don't get the order of brown versus black on the answers. The order of bears is how it is on the cladogram. Okay, sorry, I'm trying to read all of your questions and also answer questions. So I'll bounce back and forth here. Okay, so let's see. So we have polar bears are highly adapted for life in cold climates around the North Pole. Brown bears, black bears, and pandas are found in warmer environments. Researchers collected complete mitochondrial DNA sequences from several populations of bears and constructed a phylogenetic tree to represent their evolutionary relatedness. Okay. And then we also have, so this top one is based off of mitochondrial DNA. And then we also have a table of amino acid differences in a specific protein called the list protein. Okay, so A, using the phylogenetic tree in figure 1, estimate the age in hundreds of thousands of years. Did we just want to go, I'm sorry, I'm rereading the question. It looks like we just wanted to go through part B. Okay, sorry. I'll skip ahead then. Construct a cladogram on the template to represent a model of the evolutionary relatedness among the bear species based on the differences in list protein sequences. So we're just going to be looking at table 1 for this, and then circle the position on the cladogram that represents the outgroup. So the good news was you could make the cladogram, so this is 2 points, 1 for the cladogram, 1 for the circle. Hopefully outgroup sounds like, you know, the guy on the outside. So if you circled this branch over here on the left-hand side, even if you had the wrong bear on that branch, you still got a point, right? So even if you didn't write anything, if you're like running out of time, you could still circle that outgroup and get a point. That's an easy point. It makes sense. The outgroup is always going to be the one on the outside, the one that's least related to everybody else, okay? So when I look at these tables, I look at a couple things. First, the title. So we're looking at amino acid differences. So what that means is smaller numbers mean that things are more closely related. Larger numbers means things are less closely related, okay? The other thing that always bothers my students is that there are empty boxes here. Empty boxes mean nothing. They don't mean anything. Don't think that the panda is not related to anybody. It's not true. The panda is not very closely related to anybody. That part is true. But the information for panda, while it's not here, is actually just located in this column instead. Okay, so this table would be redundant if it was filled out in its entirety, because we have the same headers for columns and for rows. So if you want to go ahead and fill in the rest of the table, if that makes you feel better, you certainly can. But just know, anytime you see a blank spot, like here between brown and black bears, it's only because the information lives down here. Brown and black bears are only 1 amino acid different. The other easy way to look at this is just to look for your highest number and your lowest number. Okay, so what I'm looking at in this table is differences, and I see that there's only 1 difference between brown bears and black bears. Okay, because there's only 1 amino acid difference, it means they're very closely related to one another. So I want to put them on this tight little V here called a sister taxon. Okay, because this V portion, this intersection, is highest to the top. It means that they're the most closely related, okay? It doesn't matter if you put brown here and black here, or black here and brown here. That does not matter because we have free rotation around this axis. So it doesn't matter the order that you put brown and black in, as long as you have them on this V together. Okay, so now we just need to figure out the other two. And it's easiest now to just figure out who the outgroup should be, who is least closely related to everyone else. So it's pretty easy to see if you're looking at this, that the panda has no close relations with anyone else. 33, 34, and 40 differences, okay? The polar bear, while it also has 40 differences with the panda, is at least more closely related to brown and black bears with only 7 and 8 differences in the amino acid sequences. So pandas are going to be your outgroup, which means they're going to go all the way to the left-hand side here, and that means then you would fill in polar bears in the middle, okay? That's an all or nothing point. So if you didn't put panda, polar, and then brown and black in whatever order you chose, you wouldn't get the point for it. So it's an all or nothing again. But 1 point for that, and then 1 point for circling the leftmost group for the outgroup. Brown and polar were closer related. Brown and polar bear, let's take a look at this. Brown and polar have 8 differences. So that's more differences than brown and black. Brown and black only have 1 difference. Yep. And again, it doesn't matter if you put brown here or black here, even though one is slightly more closely related to the polar bear because we have free rotation around that axis. So you never need to stress about that or think that you need to get that too particular. Was 2018 easier? I think every year has its strengths and weaknesses. It honestly just depends on what you feel good with and what you don't feel good with. Number 2, we can take a quick at. Yeah, the brown and black need to be on the first V all the way to the right. How would you go about managing your time? So as I said in the beginning, I always go from short to long just because then I can spend whatever time I have left on the long questions, and they deserve a lot of time. But I want to make sure that I get my easy points from my short questions first. I don't want to be worried about not getting to those questions. So I always go from short to long. I usually go from question 8 to question 1. You at least want to tackle 3 through 8 first. I will go through, this second one. Okay, we only have 8 minutes left, so honestly, I don't think I'm going to be able to go through most of this. But let me try to get through some of these other questions. This is a hard one. There's a lot of tricks to it, which I would be more than happy to go through, but I don't think we have enough time to do it justice. So I want to honor some of these other questions that people have been asking. And if we do have time, I'll pull it up at the end, I promise. Okay, so homologous versus analogous structures. Homologous, same structure, different function. Think like human arm, whale arm, cat arm. All of us have the same bone structure from a recent common ancestor, but we're all using those appendages differently. Like a whale and a human and a cat all use their hands differently. That's homologous structures. They come from divergent evolution from a possible common ancestor. So Savannah answered this question really perfectly. Analogous is going to be same function, different structure. So think like bird wing and insect wing. Insect wings don't have any bones, they don't have any blood, they're not doing anything that bird wings are doing, they both fly. Those are usually from convergent evolution, meaning that it was evolutionarily beneficial for an insect to fly and a bird to fly. So they both evolved the ability to fly, but separately, not from a most recent common ancestor. Okay. Strategies to figure out what type of pedigree one is. Yes, oh my gosh. I had a whole big thing on this. Okay, so I'll go through this briefly, and I can type this up, or you guys can be scribbling while I'm saying this. Okay, first thing that you want to do is see if males and females are about equally affected. Okay, so you want to count up how many circles are affected, count up how many squares are affected. If we have pretty much equal, then that's going to be an autosomal trait. Okay, if we have very much unequal, like we have a lot more males being affected, then you know it's a sex-linked trait. That's an easy differentiator. So that's always my step 1 when looking at a pedigree. Count up how many males and females are affected and see if they're about equal or unequal. Next, you're going to go ahead and figure out if we're skipping generations. If we're skipping a generation, so one of the lines on the pedigree has no affected individuals, then it's a recessive trait. If we're not skipping generations, then it's a dominant trait for the most part. Yeah, affected ones are the black ones, but they'll always have a key as well. So those are that's like my high level. Mitochondrial, really quick, this like never comes up, but mitochondrial is going to be where, so we get 100% of our mitochondrial DNA from mom because it's in the egg, not in the sperm, the mitochondria. So if a mom is affected and all of her offspring are affected, then it would be mitochondrial. Usually heterozygous. Depends on if it's a dominant or recessive trait. If it's a recessive trait and you're heterozygous for it, you're a carrier. Usually they're not half shaded. Usually they're not shaded at all, but sometimes they are. But yeah, if you're a carrier, then sometimes it's half shaded, but oftentimes not, from what I've seen in the AP exam. But if you're heterozygous for a dominant trait, then you'd be shaded in just like everything else. Okay, parts of ecology that are most important to know. I would make sure that you review the relationships. So mutualism, both organisms benefit. Commensalism, one benefits, one's unharmed. And parasitism, one benefits, one is harmed. So relationships like a predator-prey graph, take a look at a predator-prey graph just so you're familiar with that. Let's see, what else about ecology is important? Again, graphs and relationships are really the key things there. So those are the things that I would say to focus on the most. I think ecology for the most part is straightforward. You should be relatively familiar with food webs. A keystone species is going to be something that's up towards the top of a food web, and when you remove it, the rest of the ecosystem falls, collapses. So that's something important to know. Invasive species take over and just like kind of take over the niches of all the domestic organisms who live there. That's an important thing to know. And just know that we lose 90% of the energy to heat each time we move up a trophic level. Mariah, great question with signal transduction pathway. I went over it on Wednesday and Friday, so I'm going to ask you to go to either my Wednesday or Friday video to watch that because I went over it both days. Oh, for transcription, there's initiation, elongation, and termination. So Zach answered that. So RNA polymerase is making mRNA. Then the spliceosome comes in and removes introns and keeps the exons. We had a 5' cap. We had a 3' poly A tail. It looks like everyone did a really, Zachary and Sophia did a perfect job of answering that for you, Sean, in case you were confused. What people do we need to know for the test? 0 people. If you needed to know a person, they would tell you about them. The only experiment that sometimes shows up that you might want some background knowledge of is like Miller and Urey. Yeah, but everything else, I mean, even them, they would explain the experiment as long as you're like generally familiar with the processes. You don't need to like come in memorizing any people, and they would never ask you to write about a person. Okay. Say, for example, on the FRQ section, it says identify one part or evidence or something, and you are not sure if that one thing works. Okay, so if they're asking for evidence and you include 3 different things and you're not sure which one is right, they'll actually just grade whatever is correct as long as you don't contradict yourself. So you're not going to earn more points, as Zachary said. If you write more than one thing, you would only not get the point if you contradict yourself. Like you say something's going to be purple, but then it's also going to be yellow. But if you say something's going to be purple, and this is a bad example, but like purple and spotted and fuzzy, and one of those things is right, you're not contradicting yourself. So they would still give you the point. So it's not, they don't just grade the first thing. No, they don't just grade the first answer you put. They'll read through the whole thing and give you any points that you deserve. During the 10-minute reading period, I would suggest that you start answering questions. Definitely. I would spend like maybe 3 minutes or 4 minutes kind of working through your free response and figuring out what order you want to tackle them in. But then I would start writing because I think the writing time is way more important than the reading time. Okay. We went over the common math questions, so hopefully I answered that. According to every reader, sorry, I'm seeing that there's not consensus on the right-hand side. According to every reader that I've talked to, they'll give you points for anything as long as you don't contradict yourself. So that's coming from the mouth of a reader. So I would trust it, but if you just want to go with one answer, that's totally fine too. Almika, we did camp plants yesterday, so you can rewatch my replay there. It looks like Zach said that. Yeah, I always say go from 8 to 1, short to long, or at least tackle 3 through 8 before tackling 1 and 2. Obviously, 1 and 2 are worth a lot more points, but students often get a lot less points on them for the time that they put into it. So you want to think of it as like a time point kind of value scheme. You want to get those shorter answer questions done first and then spend more time on the long ones. Okay. Water potential, I would not stress about. I would not make that one of your topics that you study late night tonight because it may be one grid in, but most likely it won't show up on the test at all. Okay, Cool. Are there any other, I'm trying to think if there's any other questions that I can answer in a minute. Bottom line, guys, you know what you're doing. Just believe in yourself. If you start getting really stressed about what you don't know, it's not going to be helpful to you guys at all, because that's when your body just kind of starts to shut down. So just believe in yourself. I'm really excited for you guys. You should be really excited that it's almost over. Please get a good night's rest. Please eat a good breakfast. Even if you don't usually eat breakfast, you should eat breakfast tomorrow because you're going to be up early and working harder than you ever have. Good luck, good luck, good luck, good luck, good luck. Take some deep breaths. You should go to bed now. And yeah, can't wait to hear about it on the other side. So great work. Don't stress about it too much. And you're the best.